Hello, students. This is Dr. DA again, and we are going to be walking you through experiment P16, which will be available by the time you watch this video here on your Canvas page. And it is not complete at the time that I'm doing this video, but this is the page that you will see. You'll have access here to the actual document that you're going to be reading through, and you can either download it or you can just read it here. Many of you should have your lab manuals with you so you can read it in hard copy there. And for this presentation, make sure that you have downloaded or have opened this document, which was prepared by Dr. Mary Perot. And just like the previous experiments, it shows you pictures at key stages of the process so you can compare them later on with your unknown. When the assignment goes live on Canvas, this video will be in here as well as my notes. All right. So if we can look at the actual lab, you will see that it involves several steps, and it's a little more complicated than the previous one. So it's been a while, but in a previous activity, you were given the analysis for group 2 cations. And remember that the strategy is always the same. You are given a mixture. You learn a few chemical reactions that allow you to separate and confirm for the presence of a particular ion. And then you are given an unknown mix with the data so you can determine which of the cations are present in your unknown. Now for this particular experiment, we're going to be using two groups at the same time, groups 3 and 4. And because of that, the flow chart seems a little more complicated. You can see it here on this page. This is page 62. Uh, and it's more complicated essentially because you are tracking essentially two threads here, two sets of ions. Group 2 here on the left, I'm sorry, group 3 here on the left, I'm sorry, and group 4 here on the right, okay? So let's do this. Let's go to a piece of paper here so we can analyze this a little more thoroughly, all right? Okay, let's start out by talking about this group 3 ions. And we're going to specifically focus on iron, which will come in both its +2 and +3 oxidation states. We're going to talk about nickel, and then we're going to talk about chromium 3 specifically, all right? So if you remember, we talked about the equilibrium of hydrogen sulfide. And we said that in solution, you can have an equilibrium between protons because of its slightly acidic activity and the sulfide ions. And those are the ones that we use to precipitate several cations in the form of sulfides. We found out and we derived this expression last time that the relationship between the sulfide ions and the hydrogen ions or protons, essentially the pH, is a reverse or inverse relationship. Uh, we said that this relationship holds. We can see that the relationship between sulfide ions and protons is inverse. So if I have, let's say, a solution at a low pH, which means a high concentration of ions, because of the inverse relationship, this is going to generate a low concentration of sulfides. And so therefore, only those cations that precipitate at very, you know, at very, very sensitive KSPs are going to precipitate. So this is what we use to precipitate the group 2 ions. These ones only require a very low concentration of sulfite in order to precipitate, and so we manipulate that by manipulating the pH. Now, if I bring this to a higher pH, in other words, a low concentration of ions, what that's going to say is I'm going to have a higher concentration of sulfites. And therefore now, pretty much both group 2 and group 3 ions will precipitate, okay? So, of course, in today's activity, you're not going to have any group 2 ions, so we don't have to worry about that. Normally, you would first do a precipitation at low pH to get the group 2 ions out, and then you would raise the pH to get the group 3 ions out. But in today's case, we're not going to do that. So what we're going to do in this experiment is, of course, remember that our source of sulfides ions is that compound thioacetamide, which we abbreviate as TAA, because that way we generate them in solution, all right? And we're going to take advantage of the conditions here in order to also separate the group 4 ions, all right? So essentially, if you look at your procedures here, going back to our procedures here, what you're going to be doing is essentially, as you can see here, you're going to start by first raising the pH to kind of like a basic, uh, you're going to start out with a step where you make the solution basic that has all these ions, and then you're going to do your thioacetamide precipitation, and that's going to separate on this side the group 4 ions, and in the precipitate you're going to have the group 3 ions. So that's essentially the first step that we're going to carry out here. Now, it's very important, as you can see, in this first step, if you read the procedures thoroughly here, back, in the actual discussion here, step by step, you'll notice that you're going to be given a group 3 and 4 mixture. And notice that you're going to add, first, you're going to add acid, and then you're going to add ammonia. The objective of this is essentially to generate a buffer. And this buffer is going to allow us to help with the precipitation. Notice that initially you precipitate the iron ions and the chromium ions as hydroxides. And then after that is when you're going to add the thioacetamide. All right. So our first step is going to be that separation, right? So let's come back here to our screen. All right. So we're going to start out by having a mixture. This is going to have group 3 and 4 ions. And then we're going to do essentially a, basic pH precipitation with thioacetamide. And that's going to separate, leaving our barium and calcium ions in the supernatant. These are the group 3. I'm sorry, these are the group 4. And in your precipitate is where you're going to have your group 3 ions in here, okay? Now, what's going to be interesting here is that actually these are going to precipitate ultimately as sulfides of, let's say, iron 2, iron 3, nickel sulfide. But the chromium is actually going to remain in the form of the hydroxide ion that is caused by the action of the basic pH. So the chromium does not go into a sulfide mode, but it is still precipitated in the form of a chromium oxide. In the typical activity in lab, you would have done this in 2 days. So one day you would analyze, let's say, the group 4 ions, and on the second day you would analyze for the group 3 ions. And typically what we do is we have you do in parallel a mixture that has all of them in plus your unknown, so you can track them in parallel. All right. If we look at the document that has the mixtures here, right, you can see that in that first step, when you receive the mixture, it looks very dark, like a dark green color, and that's pretty much because the essentially because your chromium ions turn to form a dark blue color, and then the nickel has a green color. So that's why you get this dark color. After the diacetamide basic precipitation, you're going to get a separation. You can see here on the left, this is the precipitate that has the group 3 ions, right? The iron, chromium, and nickel, also the iron 3. And then over here, you're going to have a semi-clear uh, supernatant that should have the barium and calcium ions. So what I want to do first is let's discuss what we're going to do with the group 4, the barium and chromium. I'm sorry, the barium and calcium ions. All right, we're going to start with that first. So what we would like to do, our goal is over here is our goal is to separate these 2 ions by forming a very distinguishable precipitate of barium chromate. And the reason why this is distinguishable is because it's a yellow precipitate in here. All right. Now, let me tell you what the issue is. And again, you know what, guys, I'm kind of going a little overboard here with explanations because since you're not really in the lab, you wouldn't experience this. But just so you understand, what are some of the things that we have to contend with when we do these analysis? And it is that it's not always very easy to separate these things. All right. So, like I said, we want to form this barium chromide. Now, the problem is that calcium also forms a calcium chromate precipitate, but it's more soluble. In other words, it takes more of the chromate ion to precipitate calcium than it does with the barium. So what we're going to do is we're going to play a trick, all right? And here's what happens. Of course, we're going to be adding chromate ions, but what happens is that if you do this in an acidic environment, all right, and let's say I have the chromate ions and I have protons in here, this generates a reaction that results in another ion called the dichromate ion. All right. And the other product is water. And let's go ahead and balance this equation here properly. And this equilibrium has a very high equilibrium constant. In other words, it is very likely that in an acidic environment, you are going to go this way. You're going to take the chromate away from the solution. So let's think Le Chatelier, all right? Remember Le Chatelier principle? Let's say that I have a concentration of acid that is high. In other words, a low pH. Think about Le Chatelier. If I'm increasing this component, that means this equilibrium is going to go to the right. In other words, basically, chromate ions are going to be removed. So there's not enough chromates to precipitate even the barium chromate. So neither barium chromate nor calcium chromate precipitate. Okay, now what happens if the hydrogen ion concentration is too low? So if this concentration is too low, in other words, we have a high pH, now what's happening is, since this component is lower, this equilibrium comes back to the left, and therefore you have a high concentration of chromate ions, which means that both barium chromate and calcium chromate precipitate, right? So obviously the strategy here is we want to make sure that we have the right amount of chromate ions in there. So we precipitate only the barium chromate, but we leave the calcium ions in solution, of course, for further analysis. All right. Okay, so let's discuss then how we're going to tackle this group 4 set of ions. Okay, let me move this out of the way here. So here's our attack. Sorry, our attack. Here is our analysis of group 4 cations. Remember that in the first few steps, right, we had done a TAA precipitation, and we have barium ions and calcium ions in the supernatant, remember? So what we're going to do now is, first, we're going to precipitate them. So we're going to precipitate them using ammonium carbonate. I think that if you read our, sometimes in some textbooks where they discuss these sequential qualitative analysis, sometimes what they do is they use a phosphate precipitation, but we're going to use the ammonium carbonate. And what it's going to do is it's going to, after you spin, you're going to have a precipitate that's going to have barium carbonate and calcium carbonate. Right? This is going to be our precipitate. Now remember, whenever you have these compounds where, let's say, one of the two ions is susceptible to pH, right? Remember that the carbonate ion is in equilibrium with its you know, carbonic acid, bicarbonate, all that kind of stuff. So if I now add acid, what's going to happen is carbonate ions are going to convert into essentially carbon dioxide gas. And you remember the sequence, right? This gets protonated to bicarbonate, carbonic acid, and carbonic acid decomposes to carbon dioxide, which basically bubbles out of the solution. Now, here's what we're going to do. We're not going to use any acid. We're going to use acetic acid, right? We're going to use acetic acid, right? Now, if you look at your procedures here, what you're going to notice is that the way this works is you cause a precipitation in ammonium carbonate, and then you dissolve the precipitate with acetic acid. And when you look at the explanation here, you'll realize that what you are doing is that you're going to be generating a buffer, okay? The combination of the ammonium ions that were in here and the addition of acetic acid is going to give you essentially a buffer. So this is going to essentially create a buffer. That's pH 5. So you can spin again. There should be no pellet anymore. You have basically solubilized the barium and the calcium ions again. But this is now at pH 5. And I believe that the procedures ask you to go use a piece, not of litmus, but of pH paper to make sure that your solution at this point is at pH 5. Because see, now, if you now add potassium chromate at pH 5, what's going to happen is your barium chromate is going to precipitate, but your calcium ions are going to remain in solution. So if after adding this reagent, I do a centrifugation, a spin, this is going to be my precipitate. This is going to be my supernatant. All right. Now, in some instances, they ask us to take this precipitate, this pellet, and do a flame test. As we mentioned in class, some ions have particular colors when you put them to the flame, and barium does that. Um, we usually in our lab, D319, don't have a lot of Bunsen burners available. So we typically don't do the flame tests. So you'll settle for that. At this point, the calcium is going to be in the supernatant. So what the procedure asks you to do is you're going to be adding oxalate ions. And that is going to produce in here a calcium oxalate precipitate that has a white color. Um, I think that if you look at your handout here with the mixtures, after you took, remember we had this supernatant here that had the barium and calcium, and then what we did was we added the ammonium carbonate and we produced that pellet of barium and calcium carbonate, and then we redissolved it in acetic acid, and between the ammonium carbonate and the acetic acid, you generate that buffer that gives you this test tube here. Now, potassium bromate, I'm sorry, potassium chromate, the reagent you're using to precipitate is yellow. So yes, everything's gonna look yellow here, so, but it's not the color of the mixture that we're looking at. We're looking for the color of the barium chromate. And you can see here the precipitate of barium chromate, right? Uh, and once you've separated that, you can take now the calcium supernatant and add your calcium oxalate. And actually, it's probably, I don't think it's calcium oxalate. Let me check on that for a moment. I want to make sure that we have the right things written in here. All right. It's actually not calcium. It is, I'm sorry, it is ammonium oxalate. That's where you're going to be adding. Yeah, it wouldn't make sense to add calcium oxalate if you're trying to identify calcium. So consider that a typo in that particular handout there. All right. So we can look at that again. Let's look at this handout. And uh, again, you didn't add calcium oxalate. You formed a white calcium oxalate. Notice that the supernatant here remains a little yellowish or colored. That's because there's an excess of residual potassium chromate. Okay. So that's essentially the way this works. Now remember that you are responsible as you go through this exercise, right? You're responsible for going to your through your manual here, or if you don't have the manual, I've put in the actual file in a Word document. And remember that you have a data sheet here on page 67, and you're supposed to essentially write down all the observations here for group 4, just as if you were doing the experiment physically. Okay? All right. Okay, so that was it for the analysis of the group 4 ions. But we still have now the group 3 ions, right? So essentially the way that works was, remember that in our first step, what we did was we started out with oops, let me get this out of here, make it a little clearer. We took our mixture and we did a separation by precipitating with TAA in a basic, right, environment. And as a consequence, we had a pellet that had the iron sulfides of both the +2 and +3 irons, the sulfide of nickel, and then chromium hydroxide. Remember, the supernatant of this process, of this step, is what we used to uh, analyze for group 4. So now what we need to do is same thing. Strategy is always separate and then confirm, okay? Let's look quickly at our flow chart here just to remind ourselves of how this works, right? Let's go here. Let's go back here to our flow chart. And again, remember that what you're going to do now is you're working here on the right side of this flow chart. So at this point, I have essentially a palette that has these precipitates in there. Obviously, they're all mixed. So what you're going to see is actually just like a dark precipitate in here, right? So if you look at that handout, what's going to happen here is what you're going to see is you're going to see this very dark palette over here, right? That we generated from the thioacetamide precipitation. So now we're going to analyze that, all right? So let's go back to our procedures here and see how we're going to do this, all right? So again, take a quick look for a moment here at the flow chart. And then what I would recommend is, again, read through the procedures just as you would if you were doing the experiment. so that you have a sense of where you're at. Notice that each step here is numbered, right? And these numbers correspond to the steps in the procedure. So if you have the flow chart on one hand and your manual procedures on the other one, it's easier to follow that way. So what we're going to do is, in the first step is, we are going to separate the nickel out of the chromium and iron ions. Then we're going to separate the chromium and the iron ions, and then we're going to confirm from them. All right? So let's go ahead and look at that. I'm going to come back to our paper here, and let's see how we're going to accomplish this. Well, it turns out that in the presence of acid, our iron and chromium ions will redissolve. The chromium ions redissolve because the acid knocks off this hydroxide and neutralizes it, which pulls the equilibrium back towards the dissolved chromium ions. The iron sulfides, on the other hand, undergo an actual reaction, all right, in the presence of acid. So what's going to happen is that if I add HCl, I'm going to end up, if I spin after that, I'm going to end up leaving the nickel sulfide in my pellet because it does not dissolve in HCl, but our chromium and our iron ions, which at this point are going to get uh, reduced purely to the +2 form, they are going to remain in the supernate. So at this point, I've accomplished, one step of the separation. Please take note of a special situation that we sometimes encounter with this step. And this is in your procedures. And again, I'm going through these things just because I'm trying to simulate or give you a sense of, yeah, this is what could happen in the lab. One of the problems we have sometimes is that when we do this step, although we would expect to have a supernatant that has, the iron and the chromium ions in there, and a pellet that has the nickel sulfide. Like it says here, sometimes the liquid is not clear and there are some particles of the nickel sulfide. It doesn't form a very compact pellet. So what they teach you here is a trick where you essentially make a filter inside a dropper or a Pasteur pipette. So you essentially stuff like a little piece of cotton to the bottom of this dropper, and then you load the dropper with your mixture here you're trying to separate, and then you squeeze it through by putting a bulb here, and essentially squeeze your mixture through that cotton filter into a clean test tube. And that way you get rid of the particles of nickel sulfide that didn't want to uh, get into the compact pellet here. Okay, so please read that so that you're familiar with that step, that procedure, just in case at some point in the future you come across something similar. Now you know a trick for doing quick filtering. Okay, now um, what we'd like to do now is we would like to take the chromium, and we would like to essentially convert it into a different form. So what we're going to do is we're going to add, again, we're going to add uh, hydrogen peroxide at a basic pH. And we're going to heat the mixture a little bit. Please read the procedures there. For example, how do I make the pH basic and all that kind of stuff. And in a basic pH, and we're going to learn about some of these chemical reactions in our electrochemistry topic in a couple weeks. What happens here is that in this type of situation, the chromate ions, the chromium ions become chromate ions. Whereas in the basic pH, the iron ions essentially not only reoxidize, but they also form a hydroxide precipitate. So if we spin this mixture, we're going to be able to separate the soluble chromate ions. And you remember that chromate is going to give you kind of like a yellow solution. You know, how bright it is will depend ultimately on, um, how much of it there is, right? And then in your pellet, you're going to have your iron 3 hydroxide solid. All right. And now what we're going to do is we would like to confirm the presence of these guys. So for the chromate, which is the result of oxidizing that chromium ion, is we are going to reverse that process. We're going to add again hydrogen peroxide, which again, hydrogen peroxide is an agent that depending on the pH of the environment can act both as an oxidizing or as a reducing agent. So this time what we're going to do is we're going to do this in an acidic pH. Again, we're going to heat the mixture a little bit, and what's going to happen is this is going to be converted again to a chromium ion. And, you know, once more, depending on how much of it there is, it's going to have maybe like a bluish type of color in the solution. But that's not going to be our confirmation. That's kind of like a hint. What we're going to do actually is we're going to add sodium hydroxide here, I believe, to reprecipitate. Remember the original form of the chromium precipitate was hydroxide. We're going to regenerate that chromium 3 hydroxide, which should be a kind of like a white or gray precipitate. Okay. So that is our confirmation. Pull this up a little bit so we can write here. This is our confirmation. Now, for the iron hydroxide that we have in this pellet, again, remember that compounds that have the hydroxide ion are soluble in acid because the acid pulls off the hydroxide ions by neutralization, and that causes this solubility equilibrium to shift towards the solubilized iron. So what we're going to do here is we're going to add acid to resolubilize the iron ions back to iron 3. And then we are going to add our good friend potassium thiocyanate. And remember that in the presence of that, the iron is going to form this complex ion. And if you remember that, we've done this before, which is going to give us a red solution. Okay, and that is our confirmation. Don't worry, I'm going to have all these notes later on posted to our Canvas place, even though essentially all of this is in that handout, right? Speaking of which, let's go ahead and look at some of the pictures here. Okay, so we had done the group 4 analysis, correct? And now what we had here was we had this pellet that had the nickel, iron, and chromium precipitates. And remember that what we did was we essentially did our acid dissolving. Here it is. Added acid dissolves chromium and the iron. So at this point, you've separated, right? You can see here is the nickel sulfide solid. It's over here. And notice that your supernatant looks a little dark, which is, just residual. So you might want to filter that to make sure that you don't have any little particles of nickel sulfide still sitting in there. At this point, remember that once we do the hydrogen peroxide in a uh, acidic pH, I'm sorry, in a basic pH, we're going to generate our iron 3 solid in the bottom, and we're going to have our chromium is going to stay in solution. We're going to separate them, and we're going to add ammonia and hydrogen peroxide. So under the presence of this basic pH, what we get is we get the chromium tube, which is going to eventually convert into this kind of light blue color. All right. And that's the one that we are going to treat with sodium hydroxide so we can reprecipitate the chromium hydroxide. I don't know there's a picture here of that. I don't think they have a picture of that here. No, they don't have it. But essentially, these are the intermediate steps over here of the chromium. And remember that we had generated the iron hydroxide, which we are going to redissolve with acid. Then we're going to add potassium thiocyanate, and this is the red color that we get from the formation of that complex. We studied this before, particularly when we did the equilibrium constant determination for this process. Okay, so again, I'm sorry, what's missing here is a picture of the chromium hydroxide being reprecipitated out of this redissolved uh, chromium and iron mixture. Okay, what's left? Well, what's left now is we have to deal with our nickel. All right. Nickel sulfide is not a very soluble compound. So in order to redissolve it, we're going to have to use a very special reagent. And it's a reagent that's called aqua regia. It's a combination of HCl and HNO3, hydrochloric and nitric acids. And what happens is you get not only the acidic effect, but nitric acid many times is not used so much for its acid properties as for its oxidizing properties. And so in the presence of aqua regia and heat, what's going to happen is you're going to produce some of the nitrate is going to convert to nitrogen oxide gas. All right. So you'll get some NO2 gas, which you may or may not be able to observe. It has like a brownish color, but you may not be able to observe it, but it might be good for you to take a look when you do this treatment. See if you can see a gas being evolved that has kind of like a brownish color. But the main thing that's going to happen here is that the excess of chloride is going to give us a complex ion that has sort of like a green color. Once more, the intensity of the green will depend on how much nickel was in there. Then we're going to add ammonia to make it basic, a basic pH. And again, remember that sometimes your instructions tell you to use just simply litmus paper. Sometimes they tell you to use pH paper because you're trying to target a specific pH. So please pay attention to those instructions. What will happen here is that the ammonia is going to replace the chloride ions, and it's going to give us a new complex. And we'll study the formation of these complex ions later in the semester. All right. And this is going to give me a new complex, which should have a kind of like more like a blue color. And once more, in your data sheet, please take note of all these different steps and the observations that you make. And then what we're going to do finally is we're going to add a reagent that we're going to abbreviate as DMG, dimethyl glyoxime. Just look it up in page 70. And this is going to give us a red precipitate. You don't have to spin at this point. This is our confirmation that nickel was present in my original mixture. Okay, let me show you what these things look like. Again, let's look to that handout with your different steps. So at this point, remember, we had separated the nickel sulfide solid from the supernatant that had the iron 3 and chromium 3 ions. We took that supernatant and we did the hydrogen peroxide sequence, right? First basic and then acidic to separate and confirm the iron and the chromium. And then with the nickel, what we did was after we treated it with the aqua regia, we got some kind of a light blue solution there. You know, like I said, how bright the blue color is, it depends on how much nickel, but it should be fairly faint. Now, what's going to happen is as you change the pH, right, by adding ammonia, you might get a brighter, darker blue color. And then once you add the DMG, you should finally get this red precipitate forming here. And that's the confirmation of your nickel. All right. That is the sequence here. Don't forget that what you want to do once more is you want to make sure that on your data sheet here in your lab manual. And again, remember, you can write these things by hand on your lab manual if you have the physical copy of it. Um, but if you don't, in your Canvas assignment box, I've put a link for you to download this Word file. So you can actually just write in here in this Word file, all right? Make sure you write all these observations here, including, you know, the iron, the nickel, the chromium, all that kind of stuff. I think it goes, it seems to be missing here a line for the chromium. So you might want to include it here where it says iron 3 and then the nickel, all right? Now, what'll happen then is, and at this point, like I said, this is not ready, but by the time you see this video, it will be ready. And it is that in your Canvas assignment box, what's gonna happen is you are gonna receive a message with a file that has an unknown, all right? So you look at step 3 over here. Remember that the way this works is you go to your grades, your gradebook page, and I'm showing here the instructor version, I'm sorry. You're gonna go to your gradebook, and on the right of this assignment, there should be a little icon that you can click, and that will give you a message and the data file you have to download. And so what you will then do is, as we've done in the past, is you will essentially go back here to the data sheet on page 68, which is your unknown. Don't forget to write down, you know, your unknown number, identify the cations in the sample. Sometimes I think in previous exercises, you got to writing down things like compounds, like iron sulfide or, you know, chromium hydroxide. No, it's asking for which cations were present in your original unknown. And once more, like I always tell you, you know, in this case here, you know, it might be good for you guys to do like a flow chart. Remember, we like to do flow charts here. So I would do a flow chart here. That's how you can follow. Now remember that the flow chart that you do in this section is not like the one they gave you for the experiment, because that was for a known mixture. And in this case, you have an unknown. So your flow chart would have more of a format of, you know, I'm going to have this step, I'm going to add this reactant, and I'm going to ask. So it's going to be like a little decision box or question box that says, do I get a precipitate? And that is going to indicate whether I go on or whether it basically means, no, that ion is not present. So I would strongly recommend you do a flow chart here. Uh, remember that any other pages that you write on the side, if they're not in here, you should upload them. For example, if you decide to do a flowchart by hand on a separate piece of paper, please attach it. In any case, it's very important always. You need to provide a rationale for the choices you made here, okay? So either a flowchart, which clearly indicates the steps and how you determined that a particular ion was present or not, or whether it's, by words, just writing out. Remember, you have to consider that you are writing this report as if it were for another person, not for me, not for yourself. So if you do any kind of explanation here, it should make sense to an outside reader. Don't assume that an outside reader knows, what you're talking about. Make it so that an outside person will be able to understand this. All right? So that is the activity, and it should post to your Canvas page. So by the time you see this video, everything should be properly assembled in there. Again, I apologize for this being a little late, but I hope you enjoy this activity. And as always, if you have any questions or any concerns, please don't hesitate to contact me via email or Canvas messaging or text messaging. Another thing that I probably will do is I might extend the due date for this since I didn't get it posted until a little late. So when you look at the assignment, you might see a slight change in the due date. Uh, I would still think, probably easier to get it done, get it out of the way, because we're going to be starting a new unit in class, and it's going to be a total shift in direction. So you might want to get this out of the way. Thank you so much, and I hope that you have a great time doing this activity. Please stay safe, and once more, if you have any questions, don't hesitate to contact me. See you later. Peace out.